Hi everyone, today I'll be talking to you about my poster. It's titled Microbes, Can You Save Us from PFAS? So you might be asking, what exactly are PFAS again? PFAS are perfluoroalkyl substances that have been found to cause negative health effects. Where can PFAS be found? Well, they can be found in different types of personal care products, food packaging, our furniture, and the big contender and example that we often see is nonstick cookware. So how are microbes actually involved in this bioremediation of PFAS? Well, bacteria and strains of fungi have actually been found to be effective in the removal, specifically in experiments that have been conducted in soil and water. Microbes are a cost-effective alternative to removal technologies such as soil washing and adsorption, which often take more time, more money, and more space. So the scale of human impact that can result from PFAS is kidney and thyroid impairments and endocrine and hormone disruptors. Bioaccumulation of PFAS is also common, which is basically when PFAS becomes concentrated more and more, the more we're coming into contact with it, likely giving you a bigger chance of having that adverse risk. Another important thing to know is that PFAS has been found to be able to transfer through breast milk. So even if a baby isn't necessarily coming into contact with it directly, it could still have that contact through their mother. So, microbes, how can they save us and exactly which ones can save us? So, there's multiple forms of proteobacteria that have been found to be somewhat effective bioremediators. So, these proteobacteria have been proven effective by using bioaccumulation to bioremediate the PFAS. They choose one to use between these, and the other research bacteria depends on the conditions available, such as the availability of oxygen, the initial PFAS concentration in the polluted area, and the treatment time that's actually available. So in the picture, you can see this is actually Pseudomonas aeruginosa, and it's important to note the visible flagella and cilia it has, which likely help it propel itself and even do some type of sensing to figure out where exactly the PFAS is. Um, as we know, soils are very heterogeneous, so this would be a very useful microbe because it can travel and sense where it needs to go. It would be almost impossible for for any other bacteria to move and find what it needs to do without some type of motility device. So how are the microbes actually performing the PFAS remediation? So microbes use their enzymes to target the fluorine atoms in PFAS compounds, and they oxidize the fluorine carbon bonds, or they can add electrons through reductive processes. It has also been found that in these chemical reactions, transition metals can adequately replace fluorine. This means that enzymes dependent on transition metals could be useful in bioremediation of PFAS. Perhaps even the magnetotactic bacteria we learned about in class, which could be used as a type of lure for PFAS compounds. So you might ask why microbes are still being studied as a remediation technology when there's still much more work to do and to be done. So this is because other methods have shown to work, such as adsorption and soil washing, as I mentioned. And they do work, but they are very expensive and impractical for large polluted sites. And the most important thing to know is that they can often result in toxic waste that could be even worse than the PFAS to begin with. So, in terms of the engineered habitat for PFAS, there's a few defining features of habitats PFAS are found in, and these can vary wildly, posing as a challenge. So, in aquatic environments, it'll be highly necessary to ensure microbes can use quorum sensing and biofilms to sense and find PFAS compounds. In terms of human management of these habitats, there will also be unique challenges to each. For example, in drinking water treatment, many other chemicals and operations will be occurring. In order to effectively and safely remove PFAS using microbes, experiments and studies will need to be conducted in order to ensure that the microbes will not interfere with other chemicals or experiments that are happening within the water treatment plant and result in even more toxic compounds. In soils, humans compound, mix, and stir microbial and chemical environments. So the heterogenic nature of the soils will dictate the type of microbes that are used for remediation. And once again, the microbes will need to be experimentally evaluated to ensure their interactions will not cause chemical reactions with other pollutants that are already existing in the soil. In terms of safety considerations for this, scientists are working diligently to determine the best type of bacteria that can be used for this, and even fungi, as I mentioned before, to degrade these PFAS compounds. However, there are many challenges with the varying forms of PFAS and the large amount of energy that's required to actually break down and bioremediate the compounds. So, this makes finding the actual microbes that are capable of performing such a task difficult, and it's likely that different types of compromises will have to be made in terms of energy, cost, in each remediation scenario. So they're using metagenomics and transcriptomics as the current technologies to evaluate microbes' ability to degrade PFAS. In terms of further research for this topic, scientists are calling for the use of combined technologies to remove PFAS from the environment in the coming years, just until the bioremediation solely completed by microbes becomes better and more effective. In wetland environments, introducing new microbes that are able to defluorinate PFAS compounds will be a goal, but further research and experiments are needed to determine just which microbes those are. And another proposed approach has been the combination of microalgae with some aerobic PFAS degrading microbes. This dream team combination gives microbes the oxygen and dissolved organic matter that they can use to speed up and enhance their metabolisms, better making them able to degrade PFAS and do it more effectively.